Thank you and hello everyone. I'm glad to present you LiMeTrack today, our lightweight biosample management platform we use in the multi-center S3c consortium. When working with shared spreadsheets for sample or metadata management, you probably all face the one or the other issue here. For example, you have to face incomplete data sets, you have type convention errors, or you have a lot of inconsistencies, for example, as some of the columns were sorted, but the others were not. So you end up in a complete mismatch of the patient identifiers and corresponding metadata. In addition, Excel spreadsheets harbor a risk of data loss or accidental change of the files, and you have no logging possibility or a dedicated user management. And probably all of you have faced the nightmare like this, where you have to decide for one of the final versions because you had no proper version control. And to face all these challenges for the S3c consortium, we have developed LiMeTrack. But first of all, let me introduce you S3c to you very quickly. So it's a German national-wide consortium with 17 project partners, and it stands for Spatial and Temporal Heterogeneity in 3 hard-to-treat cancers. And as this acronym suggests, we are working with multi-omics technologies to study intratumoral heterogeneity. And to do so, you first of all need scientists, and we have involved more than 100 researchers in the project that analyze patient biosamples. And currently, in the roundabout middle of the project, we have recruited more than 700 biosamples that are processed in more than 10 different processing labs. And from there are sequenced with a variety of bulk and single-cell or spatial techniques. And then all the data end up at an HPC system, where data analysis of different project partner sites have to access and work with the data. And you could imagine that at each stage, from the patient surgery to the sequencing data file, we generate and have to manage a variety of different metadata. And to do this in a structured way, we have developed LiMeTrack, which is an all-in-one solution for biomedical research projects, from the surgery and biosample recruitment part over the wet lab experiments to the sequencing data file. And we have designed it in a light way, so that it's especially designed to serve also projects that do not have the support of a dedicated data steward. LiMeTrack is password protected and works with users assigned to different user groups. And after login, the user starts at the project landing page, where all the project-related resources and important meetings are linked and explained. Sample entries can then be made on the sample tracking page, and as upload in batches is the most convenient way, we have placed the file upload option on top of the submission form. You could upload your files in a CSV or in Excel format. The batch upload, or next to the batch upload, you could also enter a single sample via using the sample entry form. And here we made concrete entry restrictions based on drop-down menus or restriction of the entries that are accepted. And before entering data into the database path, we have to do a validation, and inconsistencies are immediately reported to the user. Our sample tracking tab serves then as the complete overview table, where you have all the data displayed in a tabular form. We have a variety of options to filter rows and columns, and patient and sample counter in the header summarizes the currently applied filters in summary to the overall samples that are recruited currently in the project. You could download a filtered or the overview of the all samples here that could be immediately feed into pipelines, for example, in SnakeMake or Nextflow, to analyze your data. All the data in the databases are feed into an interactive dashboard where we display interactive overview figures that could also be exported in PNG format. And based on the project needs, you are also able to incorporate more figures based on the metadata stored in the platform. So overall, LiMeTrack addresses a plethora of potential failure points you could encounter when working with shared spreadsheets. It increases research transparency and ensures data integrity, and thus allows a lot workflow optimization, and is an important step for modern biomedical research projects. If you want to learn more about LiMeTrack, we also have a poster, so please visit us at poster A122, check out our repository, or, for example, test LiMeTrack by yourself. And thank you very much. And I'm happy to take questions.